So today's lab is on the acid-fast stain. So are we all seeing the handout?
Yeah, we can see it.
So the acid-fast stain is also known as the Zeal-Nielsen stain. It's a staining procedure that's only used for two groups of bacteria, Mycobacterium and Nocardia. But we don't care about Nocardia in this course, so I'm just going to mention it, but we focus primarily on Mycobacterium. And so this is a differential stain. We can differentiate acid-fast bacteria from non-acid-fast bacteria. As I said, we're going to primarily focus on Mycobacterium. There are many different species of Mycobacterium, including many pathogens, but the one that we use is Mycobacterium flei, and this is misspelled on this handout. Mycobacterium flei is P-H-L-E-I, and that's a harmless form of Mycobacterium that we use in lab. So our primary stain is going to be carbofuchsin. And so carbofuchsin is a fuchsia color. And for all the guys in lab who don't know what fuchsia is, it's kind of a hot pink, purplish pink, reddish purple color. And it does penetrate into the thick, waxy wall that Mycobacterium has. So Mycobacterium is special. It has a unique cell wall. It's very thick and waxy. It contains mycolic acids. And so the carbofuchsin stain can penetrate, but it needs some help. So we actually heat the stain to make it penetrate. And then it gets stuck to that thick, waxy cell wall. So when we decolorize with the acid alcohol, the carbofuchsin sticks. And then everything that's not acid fast, we have to counterstain with methylene blue. So virtually everything is blue. Only Mycobacterium and Nocardia show up as that red fuchsia color. So you would make your smears, and in this case, we actually will modify the smear procedure for the Mycobacterium. We're using the Mycobacterium and E. coli, and so of course the E. coli has to be acid-fast negative because, as I said, there's only two groups that are acid-fast positive. Everything else is going to be negative. So E. coli was our negative, and Mycobacterium flei was the positive. And so you make your E. coli smear the same way you'd always make a smear. You touch, you look, you see something, you stop. And that way you get a nice thin smear. But with the Mycobacterium, that doesn't work because that thick waxy cell wall is very sticky. And so you can't touch and look because nothing will come off because it's so sticky. So this is one time when you can just take a loopful of Mycobacterium, start mixing in the water drop immediately. Just keep on mixing and then tap, tap, tap the loop because Mycobacterium is clumpy. And so you want to try and break up the clumps as best you can. You can't break them all up, and that's fine because the large clumps are something that's easy to see when you go to look at it on the microscope later. But you will also see that you have separated individual cells. So with the Mycobacterium, there's no such thing as a slightly cloudy smear. So you take a loopful of Mycobacterium and start mixing immediately and try and break up the clumps. But it's going to be clumpy, and that's fine. You still dry your slides as usual and heat fix them before you start staining. So you go to the sink. You go to the hot plate. So we use a special hot plate because we need heat. And you put your smear on the hot plate, and you put a piece of filter paper, or we used to use sponges saturated with carbofuchsin, but you use a piece of filter paper, and then you saturate that so that the smear stays nice and wet because you're going to be heating it for 5 minutes. So you need to make sure that it's nice and wet with stain. Another way to do this is very messy, which is to put your slide over a beaker that's has boiling water that's steaming hot, and that way that helps to heat it, but that's so messy. We just use a hot plate, and the hot plate gets very messy too, but at least it's not quite as messy. And so you leave it there at least 5 minutes, and then you'll carefully remove the filter paper and put that in the trash, and then go to the sink and rinse off your slide. And then you decolorize using acid alcohol. And you would add 3 to 5 drops and then rinse your slide. And the idea is that it will remove the excess carbofuscin from the mycobacterium smear and hopefully all the carbofuscin from the E. coli smear. But if your E. coli smear is too thick, it will hold on to some of the carbofuscin. So that's why you need a nice thin smear for E. coli, just like you would normally make. So you decolorize it with acid alcohol, rinse it off, and counterstain with methylene blue for about a minute. Rinse that off and then blot the slide dry. Now, the problem, when you put these slides on a hot plate and you really cover the smear with carbofuscin, it gets underneath the slide and all over the hot plate. And so you have all this dry crystallized stain on the bottom of your slide. That you need to clean off. So it's okay to wipe the bottom of the slide. You can use the 95% ethyl alcohol or the lens cleaning solution we have, and that'll clean off the excess stain from the bottom of the slide. On the top, you can't touch that, right? Remember, no wiping the top, but you will need to clean the bottom of the slide because they get very, very dirty with all the stain that's all over the hot plate, especially after the first few students have done theirs. So you do need to clean the bottom, but not the top. And then you'll look at it under oil, and acid-fast positive bacteria like Mycobacterium have this kind of fuchsia color, this kind of hot pink, purplish pink color. And the E. coli will be blue because almost everything will be blue. Now, you do have clumps, and that's normal because, as we said, those thick waxy cell walls are very sticky. So you do have clumps, which makes it easy to find in the microscope. When you zoom into oil and you look around, you will see that you have separated individual cells, and you can see them here, here, here. And so you can see the rod shape of the mycobacterium. It's not always that easy to see because the cells do stick together a lot, but somewhere on the slide you will have separated individual cells, and you can see that they are rod-shaped. Now, if you were doing a sputum smear, and sputum is mucus from the lungs, if you did a sputum smear and you did the acid-fast stain on it and you saw anything fuchsia in it, that's bad, right? That's going to be a positive test result for tuberculosis, right? Because Mycobacterium tuberculosis is the genus and species, and we do use the word tuberculosis for the disease as well. You should never see anything that's acid-fast positive in a sputum smear. Everything should be blue. Epithelial cells in the mucus, any miscellaneous bacteria in the mucus, everything should be blue, right, from the lungs. You never want to see anything, right, that's fuchsia color, because that's bad. Now we have some other mycobacterial diseases, like Mycobacterium leprae. Anybody know what disease that causes? Leprae? Historic, especially religious. Leprae?
Is it leprosy?
Leprosy. So leprosy is caused by a mycobacterium. And of course, it does a lot of damage to your skin and tissues, and it looks really nasty. And there are people that today still get leprosy, but it is treatable with antibiotics. Now, the good and bad about that thick waxy cell wall, it is hard for many antibiotics to penetrate that thick waxy cell wall and kill those bacteria. And members of the genus Mycobacterium do tend to grow slowly. So if you're treating tuberculosis, let's say that you were diagnosed with active tuberculosis. You have to take multiple antibiotics for 8 to 12 months, so for up to a year, every single day, in order to kill those mycobacteria. And the reason why is it's hard for the antibiotics to penetrate the cells. It's also those bacteria grow slowly. So you need to take antibiotics for up to a year in order to treat TB. And then once you're cured, you can turn right around and get infected all over again, right? There's no immunity to it. So that's one of the problems with treating TB. And especially since, people tend to be, poor who get it. And so it's hard for them to get the antibiotics and take them every single day for a year. They do have programs where they actually drive out, meet with the TB patients wherever they are, in their homes, on the street, wherever they may be, and actually hand them the antibiotics and watch them take them. Because a lot of these people have problems with drugs and alcohol. And so if you give them a year's worth of antibiotics, they're probably going to sell them so they can buy the drugs that they really want. So treating TB is very expensive and very time consuming. And unfortunately, we now have strains of TB that are completely resistant to every antibiotic. So we really don't want to go back to the pre-antibiotic era, because what they used to do for TB patients is they'd send them out to the country. So they get a lot of fresh air, good food, rest, and hopefully they recovered. So that's what we had to do before we had antibiotics. So we really don't want to go back to that, but there are strains of TB now completely resistant to all antibiotics, and that's not something we want. So, any questions on the acid-fast stain? No. Well, we do have a video to watch on this, and I did use the YouTube link instead of the video that's embedded in the Blackboard, just so we can use the closed caption. Now, the problem with captioning, if you've ever watched videos on YouTube and you've read the captions, the automatic captioning is lousy. It does not do a good job, especially when it comes to scientific terms or technical terms. And you're going to see that in this video where they refer to Mycobacterium as Michael bacteria. So Michael, as in the name Michael. And you'll see some other weird things like mycolic acids. They call my colleague assets. So, the translation when they caption is not very good. So you will see some weird things on the captioning. At some point, they do sometimes say Mycobacterium correctly and also mycolic acids correctly. But for most of the time, it's Michael bacteria and my colleague assets. So, but can we all see the YouTube screen?
Yes.
Yes, so I'm going to turn down my speaker so you don't get that feedback. So.
Hi, my name is Sharon Chen, and I'm a pediatric infectious disease physician at Stanford University. In this video, I'm going to introduce you to a unique bacteria called mycobacteria and show you the relationship between the cell envelope of mycobacteria to other bacteria. Their learning objectives are to distinguish the unique features of the mycobacteria cell envelope and compare to other bacteria, to describe the role of mycolic acids and other lipids as structural components of the mycobacterial cell wall and as bioactive virulence determinants, and to recognize that mycobacteria cannot be stained with the usual gram stain because of their unique envelope, and special stains are used. Before we start talking about mycobacterial cell envelope, you might be wondering why we should care about this particular bacteria. Mycobacterium tuberculosis is a causative organism for tuberculosis. Tuberculosis is responsible for more deaths through history compared to any other infectious agent. About 1 billion people worldwide have died from tuberculosis in the past 2 centuries. The mycobacterial cell envelope has no outer membrane, so it is most closely related to gram-positive bacteria. Remember that bacteria need to protect their contents with multiple layered envelope. The figure in the slide shows you the cell envelope of a gram-positive bacteria with its thick peptidoglycan layer decorated with teichoic and lipoteichoic acids. In contrast to gram-positive bacteria, the peptidoglycan layer of mycobacteria is not as thick, as you can see in the figure. And the most unique characteristic of the mycobacterial cell envelope is its lipid composition. Multiple unique lipid-based molecules compose a large part of the cell envelope, creating a thick waxy coat. You might imagine the cell envelope to look like this massive wax from lots of melted candles. These 3 figures show you how the mycobacterial cell envelope compared to gram-positive and gram-negative bacteria. They all have an inner membrane. They all have a peptidoglycan layer, but the thickness differs. Mycobacteria's layer is not as thick as gram-positive bacteria, but thicker than gram-negative. Neither mycobacteria nor gram-positive bacteria have an outer membrane like gram-negative bacteria. And only mycobacteria have a unique lipid layer. Now let's discuss each layer of the mycobacterial cell envelope in more detail. The inner membrane or plasma membrane is consistent with all the other bacteria. As I said before, mycobacterial cell wall is composed of peptidoglycan, but there's also another layer called arabinogalactan. Essentially, this is a layer of sugars, a polysaccharide polymer specifically made of D-arabinose and D-galactose. Arabinogalactan is linked to peptidoglycan and to the lipid layers. This layer provides additional strength to the cell wall. The cell envelope is also composed of unique lipids, and some very important ones are called mycolic acids. These are long-chain fatty acids covalently linked to the sugar layer, arabinogalactan. As I mentioned before, these lipids form a thick, waxy coat that has low permeability. The waxy coat also allows mycobacteria to resist drying and chemical disinfectants. In addition to mycolic acids, the cell envelope is also composed of a variety of other glycolipids that are non-covalently bound. These glycolipids can be released from the cell envelope to interact with other membranes or receptors on host cells in a variety of ways. For example, some of these glycolipids are PAMPs, or pathogen-associated molecular patterns, and can be recognized by toll-like receptors initiating an innate immune response. In short, these glycolipids are bioactive. Mycobacteria can use these glycolipids to regulate the host immune response. And there are lots of these glycolipids, many of which are referred to by abbreviations. I listed some of the more common ones so that you can recognize them as glycolipids. Because the lipids have such low permeability, mycobacteria also have porins to move essential molecules into and out of the cell. In addition, there are other proteins in the cell envelope with a variety of functions. The cell envelope structure is important in staining bacteria. For example, the components of the cell envelope determine the gram stain, positive or negative. This is also true for mycobacteria. However, the unique lipid layers of mycobacteria resist the gram stain. It can't penetrate the waxy wall, so a different, more concentrated and lipid-soluble dye needs to be used. The image shows you what happens if you try to stain mycobacteria with a gram stain. The arrows point to their ghost-like image, which are quite difficult to see. Thus, a different dye is needed. The most commonly used dye is called carbofusion, which is a mixture of phenol and basic fusion. It's that red-colored dye that you see in the tube. Carbofusion is lipid-soluble and can bind to mycolic acids. Heat is applied to increase the penetration of the dye into the cell wall. Although it is initially difficult to penetrate dye into the lipid layer, once it has penetrated, the lipid layer strongly retains the dye. This allows a decolorizing step with acid, alcohol, or sulfuric acid to wash away everything else. What remains are the mycobacteria stained with carbofusion. They are acid-fast, and this is why the stain for mycobacteria is called the acid-fast stain. I have mentioned mycolic acids and other lipids multiple times, so I hope you are getting the picture that these are very important. In fact, in terms of the cell wall, lipids account for 30 to 60% of the dry weight. Furthermore, for Mycobacterium tuberculosis, almost 1/3 of the genome is solely devoted to lipid synthesis or metabolism. Thus, it would be correct to think that mycolic acids and other lipids are not just structural components of the cell envelope for mycobacteria. They are also virulence factors that can activate or suppress the immune response. For example, lipids can serve as receptors for invading macrophages and dendritic cells. Lipids can prevent phagolysosomal maturation, allowing Mycobacterium tuberculosis to survive inside macrophages. Lipids can mask PAMPs on the cell wall so as to avoid toll-like receptor recognition, evading innate immunity. Lipids play an important role in eliciting pro-inflammatory cytokines that initiate granuloma formation. Lastly, lipids enable mycobacteria to escape the adaptive immune response by suppressing dendritic cell maturation and T cell activation.
Any questions or comments? No. All right, so I'm going to stop the lab recording now, and then in like 5 minutes, we'll just sort of talk about the lecture exam. Okay?
Okay, so should we stay on here or should we leave?
Yeah, stay here.
Okay.
I'm just going to stop the recording.